short reads came up. However, these reads can be produced very cheaply in huge quantities. And it turned out that this overlap graph approach is unable to work for this huge, large amount of reads, and it basically disappeared overnight in favor of the deep brain graph approach that we discussed yesterday. So from 2007, people assembling genome almost exclusively using deep brain graph. And then something happened. New technology, long read, generated by companies like Pacific Biosciences and Oxford Nanopore came up. They're 100 times longer than 11 reads. But they're long, but inaccurate. They have huge error rate. And there was, once again, overlap approach took over because the brain graph approach that I described, it's mainly designed for very accurate reads. It's based on the assumption that you know exactly which chimeras are present in the genome, or maybe with small errors. And that's why people actually believe at this time that overlap approach is the only approach to assemble these reads. Until my former student, and also Sean actually discovered this student, Misha Kalmogorov, demonstrated that the brain graph approach is actually perfect for assembling reads, even with huge error rates. Okay. And is it possible to remove this, or it doesn't work?
Let me.
Okay, great, thank you. And then, just 4 years ago, there was another breakthrough. People finally learned how to generate long and accurate reads. So they started generating reads that are 15,000 nucleotide long and very accurate, even more accurate than luminary. You would think that at this point, these reads are perfect for the brain approach because they're accurate. However, but now this doesn't work. I can.
What doesn't work? This one doesn't work.
Now works. Okay, good. Yes. However, in 2022, the first human genome was still assembled using the overlap graph approach. And only recently, 2 years ago, this is my former student, another former student, Anton Barkevich, demonstrated that the brain graph is actually working very well for assembling long reads. So there were many algorithmic developments just in the last 2 years, and I'm going to talk about this. So I already mentioned yesterday that the brain graph has advantages of overlap graph, and the telomere to telomere consortium also came to this conclusion. So why my former student was erased on the idea of the brain graph? One of them is using overlap graph, and another is using the brain graph. Now let me tell you the story. 12 years ago, I went to St. Petersburg to form a lab in Russia focusing on bioinformatics. The only problem was that St. Petersburg at this time looked like a bioinformatics desert. So there was practically no bioinformatician I could find at this time. And that's why I gave a few lectures in St. Petersburg to talented mathematicians and computer scientists like you. And I basically hired them without any knowledge of bioinformatics to a new lab. So they were like you sitting in this room, they didn't know anything. By the way, you know that Sean actually studied at St. Petersburg University. His photograph of this Peter's a great statue. Anyway, I came to St. Petersburg, and the first thing we did is bioinformatics bootcamp, because these people didn't know anything about bioinformatics. Not unlike we are trying to do here, but it was long. If a couple of this bootcamp continued for a couple of months, and I gave them arguably one of the most challenging bioinformatics problem, genome assembly from single cells, which has its own difficulties that I'm not going to talk about today. And amazingly, without 1 year, they developed assembler space that is now the most widely used assembler in the world. And the leaders of this project were exactly the same person, Anton Vankevich and Sergey. So they published this space paper, and there are many variations of assembly program, assembling viruses, assembling bacteria, assembling microbiome, and they developed all kinds of variations of space for these tools. And it looks like having this assembler for short reads, it should be even easier to do it for long reads, because long reads span more repeats. So at the first glance, it looks like we didn't need to do anything, but just scale our space assembler, change one parameter. Instead of k equal 215 nucleotides, change it to 15,000 nucleotides and run it, because it ran so successfully in tens of thousands assembly projects for short reads. So my next topic, why do we actually need a new assembler? Why there was an explosion of new assemblers in the last 3-4 years by many groups. Okay. Who knows how to construct De Bruijn graph from the genome? Please raise your hands. So some people do not know. Given a genome, how do we construct De Bruijn graph? Let's slow down. Let's ask somebody to tell us how do we can, given a genome, how do we construct De Bruijn graph? Let's ask Yep. Yep is here?
Yeah.
Come over. So if I give you a genome,
Yeah.
how do we construct, I give you a genome and parameter k-mer size. How do we construct De Bruijn graph?
The green graph.
De Bruijn graph.
Okay, so as I know, I will try to produce the k minus 1
And so we will
Yeah.
construct nodes
node.
that correspond to one k-my values, right?
Yes.
And how do we start connecting these nodes?
So they are overlapping between the previous k-mer and the next k-mer. But they will be overlap k minus 1.
So these nodes will be k-1 mer, right?
Okay.
And we are given a genome. So we extract all k-mers from the genome, right?
Yes.
Suppose this node is AT and this node is TA. In what case we will connect them by an edge?
One action is T at the end, T at the end.
So independently of the genome, we will connect them. You need to look at the genome to decide whether we have this edge or not. So we have a genome, and we want to construct De Bruijn graph of this genome, right?
Okay.
Do we need to look at the genome to decide whether to draw this edge or not? Let's say this edge will be TC.
Do I need to know the genome?
Yeah, do we need to know the genome?
Uh,
Presumably you need, because you construct the brain graph of genome, right?
yes. But I need the information. How many AT or how many TC? So I'm having how many edge coded from AT to TC?
Okay, I'll give you a genome. For example, genome will be ATGG. Let's say this circle. So you know what, let's do this. ATGCAT. So for the genome, how would you construct the graph?
Oh, okay, I tried.
Let's remove this.
Uh,
What will be the nodes?
I'm trying to count the number payment.
No.
Uh,
And 18, okay. Yeah.
Yes. And then I try to, and maybe I don't think and try to connect it. 18, 20. TD, oh wait. Okay. Yes, so that's actually
Yeah.
good.
Yeah, so you will have, for every streamer here, you construct an edge, right?
Yeah.
That connect its prefix with its suffix. So there is edge for this streamer, ATG, for this streamer, GC,
Right.
for this streamer, GCA, and for this one, CAT. Perfect.
Okay.
Yes, thank you.
Okay.
Okay. Who now knows how to construct the Hilbert graph? Okay, more people. All right. Anyway, so given genome, nodes in our graph are simply all k minus 1 maps in the genome, right? And edges, all pairs or consecutive k minus 1 maps. For example, AT and DC are consecutive 2 maps, and that's why we make a edge from AT to TC. We go across the genome and simply fill the edges. Is it clear? Any questions about this? Good. So each edge is essentially a k map. Each node is a k minus 1 map. Okay? Now, this is how to construct the Bioinformatics graph from reads. But how to construct the Bioinformatics graph, I'm sorry, this is how to construct the Bioinformatics graph from genome. But how to construct the Bioinformatics graphs from reads? We don't need to change anything, it's a general notion. This time, we simply define the Bioinformatics graph for reads as a Bioinformatics graph of a virtual genome, where each reads represents a separate chromosome. We simply, in the same way, we construct all k minus 1 mirrors from reads and connect them by k mirrors that are consecutive k minus 1 mirrors in reads. Okay, so there is nothing new here, it's the same thing. And there is alternative definitions that we discussed yesterday, that another way to think about different graph is given the genome, generate a path of all k minus 1 mirrors, like here, and then simply start gluing identical k-mers. In the identical k minus one mers in this path. For example, here are all k minus one mers, and I indicated identical ones by the same color. Let's say J is the same color. And what I'm doing, I'm gluing identical nodes, identically labeled nodes. After gluing J, I will get this. After gluing AG, I will get this. What I will get after gluing all identical K minus ones? There are many more, like I need to glue this and this, I need to glue this and this. So there is this gluing operation. I will get exactly what I showed before. Okay. So this is different levels. Now, we will be talking, so this new technology that generates long and accurate reads is called high fidelity reads or hi-fi reads. So we will be talking about hi-fi reads. And since they're so long and so accurate, then we can take k-mer parameter to be large. For example, we can take it to be equal to 5,000, which means since most repeats are shorter than 5,000, it means that we can breach most repeat in the genome and hopefully just construct the whole human genome by a push of a button. Let's see how realistic is this. Now, our first obstacle is this. Let's estimate the size of the Boolean graph for the human genome. So human genome is 3 billion nucleotides. If you want to construct different graph of the human genome for k-mers and k equal to 5,000, then simply storing all 5,000 parent human genome requires enormous memory. We are not able even to store it on computer, so we cannot do much. Right? So talk to your partner and come up with a way to nevertheless store this data. How would you change this object so that you can fit it in the memory? Discuss with your partner. Here's the first hint. The first hint. is the second hint. And is the third hint. If K is large, instead of these three nodes, can you substitute them by this without losing information? Right? You didn't lose any information. So you compacted this representation into this one. Now, this non-branching path has length consists only of two edges. But in the real human genome, non-branching passes will have lengths of thousands and some even million edges. When you compact them like this, it's a huge saving in memory. Therefore, what we can do instead of this graph, we can generate condensed different graph that has exactly the same information, but represent every non-branching path as a single edge. We don't need to write TA here. We can write this, and TAs automatically can be derived from this. So this graph has roughly K times less memory. And in fact, memory to store this graph practically doesn't depend on K. We solve the problem. Or at least finally we can store the whole different graph, and then we are able to do some algorithms on this. Is it clear? Okay. So our goal now is to construct a condensed different graph. The way to construct it is first to construct a different graph, as we done here. We know how to do this, and from this different graph, we can 3D condense non-branching passes and generate condensed graph. Would it work? It won't work because this graph is huge. We cannot possibly generate it, right? So what we need to do, we need to construct this graph without intermediate step of the graph. That's actually not easy, but this problem was solved 7 years ago, and in this paper. And we will discuss today the ideas from this paper. But that's not all. This problem was solved for the genome. For constructing condensed different graph for the genome. Constructing condensed different graph for reads is more difficult, but we'll also solve it today. Okay? This is the goal, but it will be a long journey. So our goal is there were hundreds of papers on building assemblers for short reads and tens of thousands of biological assembly projects based on this paper. We just need to increase the length from 115 to 15,000. In classical computer science, if you study algorithm, it's not even a problem. Nobody would bother to even mention this. But in real life, in real bioinformatics, it requires completely new ideas because ideas developed here do not work anymore. So that's once again a reminder for you that you should look at the scale of the data, scale of n in O of n, and scale of constants in this. So when we started, it was an open algorithmic problem, and I will describe how we try to solve this. We need a number of new concepts to solve, new algorithmic concepts, and we will try to go through them slowly, so interrupt me when something is unclear. But before we go into this, we should always remember about biological application, why we are doing this. why it was so important to completely sequence human genome so the era of complete genomics started. So in the original draft human genome with many gaps, the most prominent gaps were centromeres. Remember these pictures of chromosome when they're connected, go like this, or they're connected somewhere in the middle to each other? These regions are called centromere and they're responsible for chromosome segregation. They're extremely repetitive. And of course, biologically important, something wrong with chromosome segregation, you have problems with infertility. And they're implicated in cancer. But nobody can possibly evaluate this association with cancer and infertility because these regions weren't available for the last 20 years. So finally they're available. There is a lot of studies in this. Another, and also they represent centromere puzzle. These regions are the most rapidly evolving regions in the human genome. And at the same time, they're extremely conservative. Like their function is chromosome segregation, which is the same all the time across all species, across all chromosomes, but they're so different between different mammals. Another area is population-wide immunogenomics. Understanding, for example, why bats do not care about many viruses that attack them. Well, maybe because the immune system has an order of magnitude more immunity genes than human immune system. But prior to the study, it was extremely difficult to sequence immunoglobulin regions. And another area, this time in the area of complete metagenomics, is studying microbiome. We have microbiome that consists of over 1,000 different species, and of course, multi-billions of cells living inside us. Their total genome is much larger than our genome. And in complete genomics, its branch, complete metagenomics, enable us to read entire bacterial genomes inside in human microbiome. So I won't have time to cover this and this very recent development, but I will talk a little bit on this just to talk a little bit more on the topic that Sean started covering, which is central dogma. So let's talk about a little bit about central dogma. But we'll start with this genocide of cisco. So there is a cancer drug called Yondelis, very expensive cancer drug, soft tissue sarcoma and ovarian cancer. It's isolated from cisco. And to get just 1 gram of Yondelis, you need to collect 1,000 kilograms of cisco. But it turned out that guilt by association, cisco has nothing to do with this. This is an active ingredient of this drug, is a peptide produced by a bacterium living in cisco. And so now, if you want to figure out what is this active ingredient, you need to sequence to figure out which of hundreds, actually thousands of bacteria living inside cisco is producing this. And the only way to do this is to get genome of this bacteria, or all this bacteria understand what's going on. So to explain how complicated it is, I wanted to return back to central dogma. DNA makes RNA, makes protein, right? Everybody knows this. So to make protein, you need to have RNA and you need to have ribosome to do protein. But can you assemble amino acids into a protein without any RNA and without ribosome? There are biologists in this room, so this is a question for you. Can you assemble a protein without RNA, without RNA, without ribosome, can you make protein? So let's ask people who know, who are biologists here. Okay, what do you say? Chemical. You mean human can do it, or a species can do, bacteria can do it. Bacteria cannot do it. Okay. Any other thoughts on this? Bacteria actually can do this. And it's one of the most pharmaceutically important processes. And this process is called non-ribosomal peptide synthesis. When a giant protein assembles another small protein without any RNA. And you all actually benefited from this process because most antibiotics are process related. Like antibiotics like penicillin and vancomycin are done defying central dogma and are assembled based on protein making the master protein. Okay? Sorry if it contradicts your biology 101 course. Yeah. And these are some of the most successful drugs. These drugs, they simply stole the drug from bacteria. Bacteria use these drugs to fight each other. They simply stole the ideas of the drug from bacteria. Yes, so that was discovered but this protein assemble peptides according to some code, right? The must be protein should have a code. Like RNA has a code to assemble protein. And when we have non-ribosomal peptide synthesis, this protein should have a code. And this code was discovered 25 years ago by Mohammed Raphael. So, and it works like this. So this amazing feat is done by protein, giant proteins that's called non-ribosomal peptide synthetases or NRP synthetases. And these are some of the largest and most complex proteins known. And these are multimodal protein. For example, this protein consists of 10 so-called A domains. And each of these A domains are responsible for adding a single amino acid to a growing peptide chain in a new part protein. If you see this protein, mini protein is growing, right? It consists of 10 amino acids. Each of these A domains partly has a code that decides what amino acid I am. Right? And finally, in the end, there is also cyclization. Usually these peptides are cyclic. Why is that cyclic? Because these peptides are like antibiotics, are like bacterial bullets that one bacteria uses to kill another bacteria. And cyclic is more difficult to destroy. It's better bullet than linear peptide. Okay, is it clear? So how is it done? What is the code for this domain to decide which amino acid to add? Here comes deep learning. There's a lot of studies today on this. Like I'm presenting you a very simplified picture. In every A domain, there are 7 positions. If you align them correctly, there are 7 positions. And amino acids of this position define which new amino acid to generate. For example, this is the most important amino acid in each domain. So they correspond to this A position, right? To this string of 8 amino acid, and this code result in adding this amino acid. This code result in adding this non-standard amino acid. And this code result in valine. Okay. Now, non-ribosomal code is not as precise as genetic code. As a result, even with this advanced deep learning method, they actually are not quite sure what is added. It may be one amino acid, maybe another amino acid, maybe another amino acid. So there are a few possibilities to add. As a result, the bioinformaticians do not know exactly what amino acid is added by this accumulation, A and M. Maybe one of them. And we need to find out what is the path. And this is a big problem today in trying to sequence antibiotics or non-ribosomal peptide, because we still do not understand well how non-ribosomal code works. Okay? Is it clear this mechanism for non-ribosomal peptide synthesis? Now, there is, of course, big challenge today is for every antibiotic we come up with. 20 years later, there is antibiotic resistance. And there are very few antibiotics actually left in the pipeline to deal with this antibiotic resistance. So there is a great need to discover new antibiotics. But with this uncertainty, there is no good way to discover new antibiotics. And that's why we have been exploring with our experimental colleague at UCSD that has one of the best in the country antibiotic discovery group, a new way to discover antibiotics. So my colleagues actually like diving at exotic location, but not just for fun, because most antibiotic largest the most rich environment for finding new antibiotics today is not Amazon, it's the sea, because it's a very rich biological system, and bacteria they are fighting, so they need to develop new antibiotics to fight diseases. So they dive, they collect material, biological sample from the sea, and then they try to sequence genomes of this bacteria, and at the same time we use technology called mass spectrometry to sequence proteins generated by this bacteria. Then we assemble this bacterial genome, then we find this NLP synthetize these giant molecular proteins that generate them, and afterwards we face two questions. First question, what antibiotic is encoded by this spectrum? If we are able to answer this question, we don't need anything else. We have a perfect method for discovery of new antibiotics. Unfortunately, nobody knows how to use it. Another approach, we can ask what antibiotic is coded by this NLP synthetases using non-ribosomal code. But you saw that non-ribosomal code doesn't allow us to give a perfect answer, because there are many possibilities. So neither this question nor this question can be answered today. Our idea is when you have this information and this information together, they complement each other and they build a bridge between genomics and proteomics. Okay? And we will be have been building this bridge for many years now. And finally, we feel we built this bridge. This algorithm bridge to the algorithm that combines this data and this data to discover new antibiotics. But when we built this bridge, another bridge collapsed. And the bridge that collapsed is actually here. Because we assume that we can assemble complete genomes. But we do not assemble complete genomes as a result. We assemble deep learning graph. And deep learning graph allows many passes to this graph. As a result, since this protein, non-NLP synthetases are some of the longest proteins known, they are broken in many pieces in deep learning graph. For example, one particular antibiotics is encoded by this subgraph of the graph, of the different graph. And our algorithm for incorporation, our bridge here doesn't work. It only works if we have perfect sequence of NRP synthetases. So that's why we were not able to actually implement this vision for metagenomic cluster discovery, because everything is broken. And that's why we were extremely excited when we saw the 2 years ago long and accurate rate came in the picture, and they are able to assemble complete genomes, because assembly of complete genome was the major obstacle here. So we tried before, we shot rates that didn't work, and then we partnered with leading anti-biotic researcher partners in Denmark, and in a single project from Sweden, we assembled hundreds of novel non-ribosomal peptide synthetases, and there is a work now to figure out how to combine all this data. But that was, this complete genomics was the major advance toward learning something new about intelligence. So I hope at this point I motivated you enough to explain that complete genomics may be important for biological application, and we'll go back to difficult material, which is how the algorithms for this work. Now, Sean, should I make break at some point?
I think a small break is good.
What?
Like a
Continue further?
Yeah.
Okay. All right. So our goal is to develop algorithm for constructing condensed different graph. Different graph is hopeless. Condensed different graph of genome. They start from genome for simplicity. So I will follow in Sean's steps. The beginning will simply follow to what Sean was developed, has developed sometime. So given genome, we want to construct condensed different graph. Nodes of this graph are called junctions. There are much smaller number of nodes in condensed graph than in different graph, right? Because we condense many non-branching classes. So they will be are called junction K-1 mesh. Sorry, that's and edge labels of this graph are called splits. So this is called split. Okay. And genome traversal of CDB graphs. So the genome actually traverses the graph. We can go ATCA, then we can go CG, and then continue, and it will pass through the whole genome. So genome traversal is basically a sequence of splits, right? When we walk across the genome, we traverse edges of the graph in certain order. Okay, and this sequence of splits spells out the genome like here. Okay. So the problem we are trying to solve, they are given a genome and an integer k, and our goal is to construct condensed direct graph of genome for this k. Shown here. How do we do this? Remember, we cannot go through the trivial path of first constructing the different graph. So how possibly we can do it? How can we even figure out the set of nodes? What's the junction in this genome? How can we find out? Let's simplify the problem a little bit. Let's assume that input is genome with a set of junctions. So somebody, an oracle, give us these nodes that form junction in the genome. So we know who was the junction. In this case, how can we construct the condensed graph? Take a minute to think about this. Anybody want to come up with this proposal?
Look at all the sequences that and with junction, starting with junction and end with a junction.
They are given all junctions, so they're given all nodes in the graph. Oracle, we don't know this, we don't know what are the nodes of the graph, but Oracle gave us these nodes. With this happen, can they construct this graph?
File possible subsequent in that you go.
Yeah, because we need to find which edges to draw.
Yeah.
Right? We need to find which edges to draw and what are the labels on the edge. How do we find them? Let me give you a hint. If you know all the junction, we can walk along the genome and find where the junctions are located in the genome. Right? So we can walk, check, is it a junction? Yes, or is it a junction? Yes. So it will be a junction. Is it a junction? No. So we know where all junctions are located.
Walk along the genome from a junction to
Perfect. And then we can walk around the genome and see what is it between every two consecutive junctions, right? What is written? And this will be exactly the edge in this genome. Right? And we know the label of it. So the algorithm with the Oracle, the algorithm becomes very simple. Simply find all junctions along the genome and walk along the genome to find the labels. Make sense? And the result is split graph, because the edges are called split, and these split graphs are identical to condensed graph. So here's the algorithm. That's it. Make sense? Thank you. But there are a few questions. This is a good algorithm, but every time you ask a question, is it a junction? You're solving the whole problem. How would you make many query checking if the k-mer from genome is one of many junctions, k minus one of the genome is one of many junctions? In real terms of human genome, how would you make 10 to the power 10 queries? Checking if it came here from genome is one of 10 to the power 7 junctions. Very slow. That would not work for our goals. Right? Even on this node, we still don't have an oracle. But even if we have an oracle, still it will not work. Okay. We need an oracle. We need, not only we need an oracle, we need an oracle that instantly answers the question, does the given k minus one represent a junction in this genome? So we immediately, when we walk across the genome and we are here, we immediately get an answer to the question, is it a junction or not? Okay. So as soon as it's done, the process becomes fast. So imagine that we have this oracle, and then if we have this oracle that answer question about k-mers, can we design a junction that instantly answers the question, does a given k minus pair represent a junction? So imagine that we can immediately answer the question, does a given k-mer occurs in genome? It's a basic question about genome, right? And using this basic question about genome, can we design a junction oracle that immediately tell us whether a k minus one there is a junction? So do you remember how you solved it? Okay, think about this. Think how you solve this, how you design this junction oracle. Say again. Oh, so, say again, what do you want to design?
And that's how we get a long genome from that side.
You, yes,
Need to compare message letters
mhm.
with the there's also a pay letters. We can do that by computing, recomputing hash. Like
Hash. We'll come to this, but the currently, you are a little bit ahead of us, actually. So, let's first focus, you're correct. But let's first focus on just simple question. Somebody immediately answers the question, does a given k-meres belong to the genome? Using this oracle, you need to design another oracle that immediately answers the question, does a given k minus one represent the junction? Okay, let's try to explore this. So, to design this oracle, we need to notice that a node in the drawing graph is non-branching if it's in degree equal to r degree equal one, right? Therefore, we can check whether a node is non-branching conjunction by simply checking the degree in the drawing graph. Right? The only problem is we cannot construct this graph. So can we do this check of in degree and out degree without constructing the graph? So let's see. K minus one layer in genome may have only 4 outgoing patches. It can be extended by A, C, G, and T, right? And only 4 incoming patches. And therefore, we can figure out whether a K minus one layer represents a junction, but asking 4 questions. Whether this k-mer belong to the genome, this k-mer belong to the genome, this k-mer belong to the genome, and this k-mer belong to the genome. After getting answer to all this question, we will know how many incoming patches are in this node. And the same for outgoing patches. Right? For example, here, we asking k-mer orbitals, whether this, and this k-mer belong to the genome. We get 2 answers yes, we know that in degree equal to. To figure out our degree, we ask k-mer orbitals whether this, and this belong to the genome. We get 2 yes, the answer is 2. So, and indeed, 18 has 2 incoming, 2 outgoing. We don't need to construct this graph. We simply communicate with the oracle. This graph is impossible to construct. We simply communicate with the node. On the other hand, for this node, which once again asks for question, only 1 yes, and only 1 yes here. So we know in degree is 1, out degree is 1. We know it's not a junction. And indeed, for TC, it's a non-branching node. And we figure it out without constructing this graph. So we can use this k-mer oracle to design the junction oracle. And thus, the only thing left for us is to design this k-mer oracle. So we are answering a trivial question. Given a huge genome, how to efficiently say whether a given k-mer belongs to the genome, right? What do you think? How do we design it? How, and usually I'm able to answer this question based on the data structure 101 course. So take a minute to suggest how would you already hinted answer to this question, but let's talk to your partner and say how you design an algorithm that quickly answers the question, given a huge string and a k-mer. Does this k-mer belongs to this k-mer? You cannot afford both with the string, of course. One position every time. Okay, any suggestion? Yes.
Store the camera into hash tables, and then when we want to certify, we want to find that camera in the,
Perfect.
so we can find
Perfect. We will explore it. But right away, the next question will be, what are the problems with this hash table?
so
We'll come to this, yeah. Thank you for your answer, we'll come to this later. We will explore your approach, but while we exploring this, recall from data structure course, what is the major limitation of the hash table? Okay. Of course. But every programmer would immediately tell, let's do it with the hash table. We will use binary hash table because hash tables with linked list won't work. It's too much memory, right? So what we will do, we will have a hash function that simply hash every k-mer to an integer in the range from 0 to n-1, hash table of size n. And it allows us to store genome in this binary hash table. It's binary, every element is a bit, by computing its hash and switching the ith bit of hash table from 0 to 1. Of course, it's not reversible, so you cannot figure out to which k-mer was hashed here, and it may result in collisions when different k-mers collide on the same machine. It's not a big problem. It's not the major problem with the hash table yet. That's the problems that you caught, but we'll come to another big problem. Okay, and a good hash table should be fast and should minimize collision. For example, we can store this genome in a binary hash table like this. Our hash will be A times Q squared plus T will be basically, we'll transform ACGT into 0, 1, 2, 3. We'll check Q equals 4, size of the hash table equals 16. So when we compute hash of ATC, it will be A times Q squared plus T times Q plus C, and it will be 13. Okay? So these are all hash volume, and we take it by model 16. These are all hash volume, many collisions, of course, but that's how we store the genome. Is it clear? Okay. And now we can try to answer the question whether k-mer appears in the genome by computing hash of this k-mer and checking the corresponding bit in the hash table. If this bit is 0, then k-mer definitely doesn't appear, right? If it appeared, this bit would be right. If this bit is one, does it mean that it appears in the genome? Not necessarily. It may appear, but it may not appear. We do not know exactly because there are collisions. So it may or may not appear in the table. So the hash table may generate false positive, which means it may classify non-genomic scammers that have nothing to do with genome as genome. It says it may appear in the genome, but it never produce false negative. It will never report a genomic camera is non-genomic. So what we now have a very instantaneous junction, but it's error control. And since this K-mirror oracle is error prone, our junction oracle that check whether K minus mirror is a junction is also error prone. It is error prone, but it only overestimates the degrees because it provides upper bound because the K-mirror oracle doesn't generate false negatives. And therefore, when we generate estimate the degree of the node in the graph, if both upper bounds for incoming and outgoing edges exceed 1, we classify this K minus pair as a non-junction. Both of the upper bounds are 1, that we classify a K minus pair as a non-junction. And if the upper bound on either the same degree or in degree exceed 1, we classify it as junction. And since we have potentially false junction, the potential junctions include both real junction and pseudo junction. For example, the same example as before. Previously, we classify a T as a junction, and we computed its in degree as 2 and our degree as 2. But with this hash table, we may have fake K false positive, right? And we estimate the degree as 3, but it's still putative junction. We still classify it as potential junction. For TC, it was a non-junction, and it remains non-junction because there are no fake kind of here. But here, GG actually was originally a non-junction, but there is a fake edge here, and it becomes a putative junction. So as a result of this, we may have some pseudo junction in addition to junction. And the question is, can we determine which K minus R identified by now error-prone junction represent pseudo junction rather than real junction? We should get rid of this pseudo junction. So this is we showed before how to construct the split graph. We identify a junction and construct split and the split formula. But what would happen if we have pseudo junction? Well, there is additional junction appearing here, which is not real junction, it's pseudo junction. As a result, there is an extra node in our graph. But it's not a big problem because we can condense this graph. And it's resulting in the same condensed graph as before. We simply need to do additional operation of condensing non-branching passes in this graph. And therefore, we can easily identify this pseudo junction and get rid of it. Therefore, what I'm trying to say, pseudo junction represents just a minor annoyance if the junction article has low false positive rate. Make sense? Now, the exercise break that you won't be able to do here because it will take a little time. Estimate the probability that a random generated kernel is classified as a genomics or false positive when genome is stored in a hash table. And here comes a major problem with the hash tables. It generates very high false positive. So pseudo-Johnson represents a major problem because the false positive rate is so large that there are so many pseudo-junctions that we are back to ground zero. So the number of pseudo-junctions is so large that we cannot construct the graph. We cannot fit the graph into the memory because there are so many pseudo-junctions with this approach, with a reasonable hash table. So it looks like we wasted time. But if you remember material from data structure course, how do you deal? What is a better data structure than a hash table that allows you to reduce genius data structure? Very simple. That allows you to reduce false positive. It is covered in some data structure courses, and it's not covered in others. So I do not know you. If you do not know this, that's fine. I will introduce. Yes.
Can we like create more records?
More what?
More records for more hash
More records.
More
Records.
instead of using a different.
Instead of using one record.
Yeah. So because each has a probability of producing false positive, but if we use more records. And just
Potentially it may grow. Potentially it may grow. But there is another more classical way that exists to address this problem, and this is called in the data science, in the data structure course, this data structure that addresses the limitation of standard binary hash table is called the Bloomfield. Was it covered in your course, in your data structure course? No? Sometimes it's covered, sometimes in our at UCSD it is covered, in some other universities it's not necessarily. So this is very useful. That actually happened in many bioinformatics applications. So I will take a liberty to describe it. And it's extremely simple. Instead of, it's probably as close to what you propose. So instead of one hash function, we will now use multiple hash functions, let's say T hash function. In this case, I will use, let's say, 3 blue, green, and brown hash functions. And I will use the same binary table I used before, the same size, the same binary table. But now, when I store a KM, it's a hash table. I will compute hash 1, hash 2, and hash 3, blue, green, and brown hash. And switch on 3 bits in this table. Not 1 bit as before, but the number bit equal to the number of hash tables. It sounds very counterintuitive because it increases the chance of collision, right? I feel more elements into the hash table. So I stored element X here. And I do it with every hammer from the genome. So now I stored element Y, there is a collision, there will be more collisions than before. Make sense? Now, when I wanted to figure out whether an element belongs to the set that I store, set of hammers that I stored, what do? I take element W, I check green hash green element is red, brown element is red, but blue element is not filled, which immediately implies that W is long in the set. If it was stored, the node 3 element would be red. So W does not belong to our set. And V, since all 3 elements are red, belong to our set. Okay? So create the stable either that the element is definitely not in the set or returns possibly in the set. And I won't be covering this, but this approach seemingly it actually at the first glance, it should be worse instead of hash table. It has way better or very low false positive values of the hash. And with this, we can actually construct construct the graph, despite having some false positive. Now, we are almost ready to describe the final algorithm. The only thing left, let's estimate the running time. So storing a genome in the Bloom filter takes with k hash function, take just one single pass through the genome. We go, we find k genome, store it in the Bloom filter, and then afterwards everything is done. Right? So the running time will be O size of the genome, K multiplied by K, because we need to look at the scalars, and multiplied by T. T is the number of hash marks. Unfortunately, since our K is so large, it becomes prohibitively time consuming. So it may seem impossible at the first glance. Can you possibly shape K from this expression? And then we will have everything. We will be able to construct the brain graph of the whole genome in minutes. But it seems very counterintuitive because you should be able to compute the hash of k-memory in all form time in fixed time, independent on the k-memory size. So think whether it's possible or not. And I think good time to make the break. Okay. Never do. About the ribosome. I then remember this, but I didn't said that. I was like,
never heard of that. They were quite strong, what, how many years? And that's so. I know it's a story, I don't think they created it. Okay, the chicks are very light proteins that are made that way, is that why? Yeah, yeah. That's very. Like another cold. It's like another cold is like. It's another. I think. I don't, I had to take action. What? Two hatch functions and then it looks like it makes it work, but it actually makes it better because you can recalculate it for everything that you know. Everybody's saying. I'll tell you, I'm still there, I'm going back and let's talk about it. By the way, I gotta tell you something. I put the data structure parts. 45 minutes. So, you know, không gian số hai số exponentially All right, let's get it. Thanks for me.
Okay, so I wanted to reiterate on that, how important it is to study while you're in university. Because when you're working on a large project, and I just started, I just scratched the surface. You probably already feel we won't be able to go to the very end. But when you go through a project like this, I'm showing you the final direction. And there were people here proposing other approaches. And it's very important to know the foundations of computer science. It's very important to know the data structure because there are many different variants you can go to. And some of them will be working, some of them will not be working. You select the wrong one, you work on implementing the wrong one, and only later you learn, well, I should go a different way. That's why you should really focus on what you're studying now. So we covered Bloom filter. I assume you didn't cover it in your class. I will cover another trick that is very popular right now. Another thing that I mentioned also in the very beginning, I would not, you're probably thinking about going to graduate school, deciding what area you should be specializing in. And bioinformatics is somewhat unique in the sense that you can start doing research right after, you can start doing research now. So these people that I showed you, Sergey Nook and Anton Barkevich, Sergey was undergraduate student when he joined the lab. Most students, when I came to St. Petersburg, I was simply looking for talent. And most of them who joined were actually undergraduate students at that time. So you will be able to immediately jump into research in bioinformatics area. Anyway, let's go further. So how can we store a genome in the Bloom filter, store a k-mer in the Bloom filter, such that the time to store every k-mer is fixed, doesn't depend on k. So how can you store a k-mer of size 5,000 in a fixed time. Right? Okay, how can it be possible? Should you look at every letter in this schema to store it? And here comes polynomial rolling hash, which is a common technique for speeding up this algorithm. Did you cover it in your courses? Did you hear about this? No? Okay. So I'm showing, Sean, I think you have somewhere here something from Dick Carr. You showed me yesterday
Richard Cup,
from this Richard Carr, yes. Is it on the wall?
it's on the wall.
It's on the wall. So this is the Richard Carr, one of the most famous computer scientists. He, Rabin came up with this extremely simple algorithm, trivial algorithm, for trivial problem. And the problem is given text, given a pattern, short pattern, and a text, right? Find where the pattern appears for the first time in the text. Well, the trivial thing is you can go through the text and check every kernel until you see the pattern, right? They propose something else. That's not the algorithm yet. So you don't appreciate the power of the algorithm yet. And so the length of the pattern is k. You compute hash of the pattern, and it became h pattern. And then you go through every kernel in the text. This stand was the kernel starting at position I of the text, this expression. And you compute the hash of each kernel in the text. Afterwards, you compare versus the computed hash of the kernel at position I coincide with the hash of pi. If it doesn't coincide, then of course pattern doesn't appear at this position. If it coincides, you have an obligation to check whether this pattern indeed appears at this position because there are collisions. You do this check, and finally, if kernel position I coincide with this pattern, we return position I, otherwise we return pattern not found. Trivial, right? Average running time of this algorithm, it's not yet Rabin algorithm, just preparation process. Average running time, length of the text, and time to compute hash of kernels. So that's what takes time at every iteration. And of course, usually to compute hash of kernel, the hash of pattern, you need the time equal to the length of pattern. So k. So the average time will be text time k. So how can you use average running time to make it fixed, independent of k? So using traditional hash function is too slow, right? Because it looks at every of k position in the kernel. We will use rolling hash.